Selection and expression of insulin in bacteria. In here, we will discuss the gene transfer into bacteria with suitable vector. Here we use insulin gene. The insulin gene is responsible for the encoding information needed to produce insulin, a crucial hormone involved in regulating blood sugar levels. The primary function of the insulin gene and the insulin hormone include: the ultimate goal is to provide a safe, effective, and reliable source of the human insulin therapeutic use in individuals with diabetes. The transferring the insulin gene into the bacteria allows for the efficient and cost-effective production of insulin, addressing the medical needs of millions of people worldwide. In 1958, greater scientist Friedrich Sanger was discovered insulin structure. There are two chains, A and B. A contains 21 amino acids, B contains 30 amino acids. So if you go over the transformation step, first of all, we have to identification and isolation of the gene of interest. So in here, we use human insulin gene as our interest. So next, strategy of the insert in the expression vector. So we use PBR322 as our suitable vector. It is more ideal for this process. So in this diagram, you can find out there are two types of subunits, A and B. This both are suitable for our further analysis, and this is the most effective part of our insulin gene transformation. Next, if you go for the bacteria transformation, so this bacteria transformation process consists of different steps. The first step, we have to keep the E. coli bacteria in ice. Then we have to grow E. coli into exponential phase. Then we have to centrifuge the pellet in bacteria. Then after that, we have to add calcium chloride into it. Then add ligated plasmid 220 micrograms into 50 microliter of the bacteria cells. Then after, we have to keep it incubation for 25 minutes. So after finishing the incubation, we have to start for the heat shocking. The heat shocking parameters will be 42 Celsius to 30 seconds. After the heat shocking, we have to incubation with the ice in 2 minutes. Then add 250 microliter of the SOC media. Then we will keep 45 minutes in shaker. After that, we have to spread the 50 microliter of our bacteria in plate. Then we have to keep it 37 Celsius and overnight. So after this overnight, you can see there are two types of colonies will found. There are white and blue. So white colonies reflect the positive results, where the blue one is described the negative results. So then we will go for the QC validation. The QC validation we have to go for, first we have to pick up the 5 of the colonies, then isolate plasmid DNA using gene tech plasmid mini replicate. Then we have to purify this plasmid DNA using thermogene tech, and finally we have to go the gel electrophoresis. So the end of this diagram, you can find out the results of the gel electrophoresis. So finally, we have to find out whether we use our shuttle is more effective or not. So here we will determine the effective concentration of the neomycin for hair cell selection using our kill curve. The right side corner, you can find out the kill curve. The kill corner is like, this is the most important part. So to find out whether our vector is like trans vector or not. Then after that, you can find out the fluorescence images respect to that. These are my reference. Thank you.